Hello, good afternoon, everyone. Um, we are going to continue to talk about lecture 6 today. I don't think we can finish it, but we will get close to finish it. Um, so let's start with doing a quick recap of what we talked about last time. Um, so here is the replication bubble that we talked about, right? Yeah, so the replication bubble is created by the helicase. The helicase is going to unzip the DNA, right? And we have other enzymes that are going to help with the process. Enzymes like gyrase, which will relieve the tension. Now, hopefully, you have the sheet that you copied down from last class, right? So you know, some of the terms that I'm talking about. So helicase unzips the DNA, gyrase remove the tension in the DNA, and we want to keep these strands apart so that they don't come back together. And for that, we need to have the single strand binding protein. So on the upper strand here, we have 5' to 3'. You should draw this out as well, because there's some new information that are going to be coming your way. And the other one is going to be anti-parallel, which is why it's going to be 3 to 5 at the bottom. Now, the enzyme that is responsible for building the new strand of DNA is called DNA polymerase 3. DNA polymerase 3. And one of the problems, it has two problems. Number one, it's not able to start the process. I told you, it doesn't know where to begin. And that's why we need to create a primer, right? Create a primer. So there's a primer right here. Okay. An RNA primer is created by the enzyme RNA primase. Okay, it's just a few nucleotides that helps it helps position the DNA polymerase 3 so that it knows where to start, right? But once you do a few nucleotides, then the polymerase 3 can continue on, and it will be basically grow all the way to the end, right? Now, if this is 5' to 3' on the other end, on the new strand, it's going to be 5' here and 3' here. Okay. Now, on the other side of the bubble, we also have to put some primer here. Right? Same situation. The primase do a few examples, if you would, and then polymerase 3 will take over, but this time is going to be growing to the right, like that. Right? Because the polymerase 3 can only build from 5' to 3'. Okay. And then as helicase opens more of the bubble, you're gonna need to have another primer here. And then the polymerase 3 will be able to go this way. And then there's another primer here. And then it's gonna go this way. And then same thing here, right? You're gonna have a primer, and then you're gonna be able to go this way. Right? So this strand is being built continuously with no breaks. That's why this is called a leading strand, right? Leading strand. On the other side, you're building it in fragments, and we call it the lagging strand. Does anybody remember the names of these fragments? You probably have it written down, right? From last class. I hope you written it down. These are the Okazaki fragments. All right. Now, interestingly, you can see, even though this, like the left side, at the bottom is being built in fragments, so it's lagging strand. But if you look at the other side, it actually becomes the leading strand, right? Because now this is being built continuously, right? And it's just gonna keep on going until the end, right? Whereas the top here, it was the leading strand on the left side, but now on the other side, you're gonna have to put more primers. You're gonna put more primers here, and then you get your Okazaki fragments. And this side is the lagging strand. So that's basically where we left off last time. Any questions before I move on? We have like a lot. I say this every class. Like every class, I'm like, hey, we have a lot to do today. So, make sure you have a good foundation of the stuff that we're doing before it, snowball into something ridiculous, right? We good? We understand why it's leading on one side and then lagging on the other, and then vice versa for the bottom strand? Excellent. Okay, so I'm gonna finish up the note here. Let's zoom in on here on this part. Let's zoom in there, and I'm just gonna put a few nucleotides here so that we can visualize what's going on. And again, these are just like random nucleotides. It doesn't really matter what you put, but to make it easier, just copy what I have here. All right. If we wanted to copy this strand to build a complementary strand, like I said, the polymerase 3 doesn't know where to start. That's why the RNA primase, the enzyme, has to come and do a few for you, right? It's like, okay, there's an A, so I'm gonna pair with a U because we're doing RNA. We don't have thymine. We have to use uracil instead. And then G, G, C, and then U like that. So this here, this is called an RNA primer. Again, nothing is new. And then you might have another primer here. Okay, another primer here. Why do you have so many primers? Well, because we are looking at the lagging strand here. The lagging strand is not built in one go. You gotta do it, in fragments, right? This is 5' to 3', like this. So once you begin, the DNA polymerase 3 will go on and keep building, right? Again, it's not a new story yet. It's still the same thing we've learned. The next thing I'm going to tell you right now is new. So pay attention. Now you're going to have the polymerase 3 here. Okay, this green circle is the polymerase 3, and then it's going to move along, right? It's going to move along, and as it is moving along, it's going to build the complementary strand. So T is going to go with A, C, C, T, A, G, C, G, G, T. All right, pretty straightforward, isn't it? Okay, but here is the new thing. Watch. Again, the green circle is the polymerase 3. So it keeps on going, it keeps on building and building and building, and then it hits the primer, okay, the RNA primer that was created by the primase. When that happens, number 3 is gonna fall off. Okay, number 3 is gonna fall off, and then DNA polymerase 1 will take over. Now, let me write it down for you. As DNA polymerase 3 approaches the RNA primer, it falls off and is replaced by DNA polymerase 1. Okay, copy that first, and then I will explain more. I'm running out of colors here. All right. So listen, right? Number 3 falls off, and then number 1 goes on. And what does number 1 do? Number 1 is gonna remove the primer. It's like, okay, I'm gonna take this away, and then I'm gonna change it to DNA. I mean, it's the same thing over here. I probably made a bad example in this case, but basically, the DNA polymerase 1 will take away all this stuff and then replace it with like DNA nucleotides. Okay, like it seems like it's not necessary, right, in this case, but actually, like over here, for example, on the left, if you take a look at the primer here, we must replace these because then I have U in it, right? So the polymerase 1 will remove the U and then instead put the T back in. Okay, do you understand? So basically, it takes all these red stuff out and then replace the nucleotides. Right. So DNA polymerase 1 will remove the RNA primer and add DNA nucleotide instead. Got it? That's all. I mean, it's not super complicated. I hope. All right. That's all the notes we're gonna take for the moment. We'll come back to it in a bit. But let's switch back to our PowerPoint for the moment. This is just a diagram that summarizes everything. You will recognize some of these things that I'm talking about, and you can take a look at the study guide. There's a diagram that looks exactly like this, and you can use it to help you study. All right. All these we talked about already, so I'm not gonna read them to you. I will read this to you. This is basically what I just told you. DNA polymerase 1 remove RNA primers and replaces them with appropriate nucleotides. Most of the time, it's just the same thing, right? Like for the A, C, and the G, they just change it to A, C, and G. Like there's no change basically. But whenever there is the U, the uracil, it has to replace it with thymine because it's DNA we're building, right? And then there is one last enzyme here called the ligase, DNA ligase. The DNA ligase is basically, you can think of it as like glue. Okay, it joins the Okazaki fragments together. Okay, joins the Okazaki fragments together to form the covalent bond connecting the nucleotides. Both DNA polymerase 1 and 3 have proofreading mechanism. It's able to check for mistakes as it is building the new strand. And if there is a mistake, it will take out the wrong nucleotide and then put the correct one in. And if the error is not corrected, then it leads to permanent change in DNA sequence, and we call that mutations. We will learn a lot more about mutation next week. Mutations are not always bad. I mean, usually it's pretty bad, okay? But then sometimes it either does nothing, or in some rare cases, it can even be beneficial. For example, some people has a mutation that makes them invincible to HIV. They cannot get AIDS. Okay, the mutation makes them invulnerable to the virus. So that's a pretty good mutation to have, right? Mutation just means a permanent change in the DNA. Okay, and that change could sometimes be beneficial, sometimes it doesn't do anything, but most of the time it's gonna cause some problems. So here is the the Okazaki fragments. You can see there are some gaps here in the strand. And of course, the Okazaki fragments is only found on the lagging strand. So you only need to have the ligase on the lagging strand. There's nothing to glue together on the leading strand. Leading strand is being synthesized, created continuously, right? So no breaks, no gaps. But of course, if you remember our diagram, even though it's a leading strand on one side, it is going to be lagging on the other. So, you're gonna need to have some ligase here, 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 and you're gonna need some ligase here, here, here as well. Okay. So that's the DNA replication in prokaryotes, in bacteria, right? That's how scientists first understand the mechanism. In eukaryotic cells, us, it's more complicated. Okay, so first, here are like a few differences between us and what you've just learned. Okay, the stuff that you learned so far, that's for bacteria, right? For prokaryotes. For eukaryotes, we have linear chromosomes. Our DNA is like, it's like a long strand like that, as opposed to circular for prokaryotes, right?
Sir, sorry, I have a question.
Yes, please go ahead.
You said the ligase it's the
It's
I
the
don't know what
glue
it
that
is. It's a
puts
glue that keeps the lagging strands together.
that joins the fragments together
Okay.
on the lagging strand.
Strands. Okay.
Yeah, is that's the only thing?
Yeah.
Oh,
Yeah,
okay.
that's it.
Okay, great. All right. So our DNA is so much longer compared to that of the prokaryotes. We have billions of base pairs as opposed to just, a couple millions. So it makes sense for us to have multiple origin of replications. Okay. So, you don't just have one bubble, you have multiple bubbles, so you can do, finish the process much more efficiently, much quicker. That's the first difference, okay? Second difference is that most of the enzymes in prokaryotes have eukaryotic counterparts. Okay, so like they have helicase, we also have helicase. They have primase, we also have primase. But sometimes the counterpart is represented by multiple enzymes. So take polymerase, for example. There are only 1 and 3 for the bacteria, right? But in humans, there are 5 different DNA polymerases. It's just because we're more complicated, right? We need to do things slightly differently. That's the only reason. Okay, so that's why like you only see 1 and 3. You might wonder, hey, where did number 2 go? Well, it turns out number 2 is only found in human, and there are other ones as well besides 1, 2, and 3. But we don't have to worry about it. We just have to know that there are multiple versions of the same enzyme in humans. Now, this last part is very important, okay? Because I'm gonna keep on talking about it later on. Not like in the coming slides, I mean. Okay. In humans, in eukaryotes in general, every time we copy our DNA, it gets shorter. Okay, so let's say you start with this much DNA, your DNA, by the way, you have like 46 chromosomes, right? So each of the chromosome, let's say chromosome 1 is this long. And then after we copy it, it gets shorter and shorter and shorter and shorter. Every time we copy our DNA, we lose a little bit of it. Of course, the question is why, right? Let me go back to this diagram and help you explain it. Okay, so in prokaryotes, the DNA is circular, right? I don't know if you remember this, but I told you a long time ago, like the DNA in bacteria, it's not linear, it's not straight. It goes in a circle. All right. Now this, I just do one strand here to keep it simple. So for example, like I mean you can still have a bubble here, right? Like a replication bubble here. Okay. But the point is this. Like you can put a primer here. Okay, and then you might have multiple primers like that, right? Multiple primers. And then the polymerase 3 will build. And then when it gets to the next primer, number 3 falls off, number 1 comes on, and then replace the red with green, right? So to speak. Same thing here. Yeah. Like that, like that, like that, like that. In other words, all the primers will ultimately be replaced by DNA, right? Because there will always be something coming up. There will always be a polymerase 3 coming from upstream, we say, from above us, from before us. And when the number 3 falls off, number 1 comes on and replace the red with the green. Okay? That's the first thing to understand. So all the reds will eventually be replaced by DNA. Now that's going to be a problem if we talk about linear DNA. Linear is straight, okay? Now I'm going to do 2 strand here. 5 prime to 3 prime, 3 prime to 5 prime, just like before, except like instead of drawing the bubble, I'm just drawing it like straight line here to make it easier. Okay, so watch. You put a primer here, okay? That's 5 prime to 3 prime. And it's okay. Right? Polymerase 3 will build the rest of the strand. Okay? But as the bubble opens more, you're gonna need to have another primer and another primer. Right? And polymerase 3 can only build in 5 to 3 direction. Like that. Same thing for the bottom. Right? You will have a primer here, and then you'll be able to go all the way to the end. You will need another primer and another primer, and then go this way, right? So listen to the explanation and see if it makes sense. The polymerase 3 will go and build a green strand, but then as it approaches the primer, it will fall off, right? I keep on telling you this. And then number 1 will come on and it will replace the red with the green. It will replace the RNA with the DNA. So that's fine. This part here will get replaced. It will become DNA. And then over here, same thing. Right? As the polymerase 3 comes, it will replace the DNA with the RNA with DNA. But we're gonna have a problem here at the end, right? This little bit here is not going to be replaced by DNA, right? This primer is not going to be replaced by DNA. Why not? Can someone please tell me why can we replace this with DNA, but not the one at the very end? What's the difference? The difference is this. We don't have a polymerase 3 coming from upstream. But this is the end of the line. We don't have a polymerase 3 coming. If the polymerase 3 is not coming, there is no signal for polymerase 1 to get on. And this part here at the end is just gonna stay as RNA primer, and it will be lost. You're not gonna copy that section of a DNA. Does that make sense? Same thing at the bottom, right? Look, the polymerase 3 will call and then fall off. Number 1 gets on, replace that with DNA. Same thing here, replace that with DNA. But this part, right? No polymerase is coming from upstream. Upstream means like above, right? Like from before that point, right? From upstream, this will not be copied as DNA. Now, why is that not, why would that not be a, by the way, this is eukaryote. Why would that not be a problem for bacteria? Well, their DNA is circle. So it just goes in a loop. Look, it just goes in a loop. And then you'll be able to replace it eventually. You just end back where you started at, right? So that's okay. But if it's linear, we literally don't have anything coming from above, from upstream, and you lost the end of your DNA, both ends, you're gonna lose a little bit of it. Okay? Any questions about this concept? Because I'm gonna this point is important. I have a few more slides that tells you why this is important. So I just want to make sure that you absolutely understand. And if not, that's okay. You can just ask me some questions. No, we all good? Really?
Well, it doesn't make sense to me. I'm sorry.
No, that's okay. But which part doesn't make sense? Can you like let me know?
Uh, it doesn't, nothing makes sense. Can I see if I can repeat it in my own words?
Yeah, of course. Go ahead, please.
Okay. And I'm probably 100% wrong. You're saying that, okay, these little red lines, is that the ligase
Okay,
that's
so
keeping those things together?
the red lines are the
Yeah.
RNA primer. Okay, the RNA primer. So let's backtrack a little bit. Let's take a look at over here. Remember, because we cannot, the DNA polymerase 3, which is the one that actually builds the DNA, right? They cannot start the process. They don't know where to start. That's why we need to have the primer. The primer do a few examples before polymerase 3 can take over. Does that make sense, that point?
Yes.
Okay, so here's the thing, right? The primer is RNA, and we are in the business of copying DNA. That's why when the polymerase 3 gets to the primer, number 3 will fall off. And then another polymerase called number 1
Will take over.
That's right. And then it will remove all the red ones here because those are RNA, right? And then it will replace it with DNA. So in other words, like in terms of colors here, right? You remove all the red ones and you replace it with green ones. So far, so good?
Yes.
Okay. Now, the ligase doesn't really have anything to do with what we were talking about. The
Okay.
ligase just connect all the, all the fragments together. Okay.
All the lagging fragments.
That's right, exactly. Now, here's the thing. If this is the end of my DNA, if this is the end of my DNA, okay, what is going to replace this primer? Nothing. Because I don't have another polymerase coming down and then telling polymerase 1 to get on and replace it. You know what I mean? It's not like over here. Over here, the polymerase 3 falls off. When number 3 falls off, it's the signal that tells number 1 to get on. Right? Number 3 has to get off and then be like, hey, number 1, come over here. You got to remove the primer here. It doesn't belong here. But that doesn't happen for the end of the chromosome, right? Because there was no number 3 coming from above. So without the number 3 coming off, a number 1 is not going to get on and replace these green nucleotides. And what ends up happening is this part just disappears. And you end up not copying the ends of your chromosome. Does that?
Is this because it's a linear?
That's right, because it's linear, right? It's straight. So nothing, you cannot loop back on yourself, right?
Okay.
You waiting for something to come down to replace the primer, but this is the end of the line. Nothing is coming down. That's why they don't get replaced. That's why they are lost. And we lose a little bit of DNA at both ends every time we copy it.
Okay.
Is that slightly better, maybe?
Yeah, that this makes a little bit more.
Okay, Great.
Thanks. I have a question for
Yeah,
you.
yeah,
I
sure.
I've been meaning to ask this question for a while, and
Yeah.
I'm not sure when was the right time to ask. But since we're talking about chromosomes from, and DNA,
Mm,
obviously that we get from our parents,
mhm.
right?
Yeah.
Why is it some people that have allergies or certain types of allergies and some don't?
Yeah. So actually, we're gonna probably address a little bit of this as well. Not everything is 100% genetics, right? That's the short answer. Right? Not everything is genetics. Even though things have a genetic basis to it, it has a lot to do with the interaction with the environment as well, right? So there is this concept called nature versus nurture in biology. Like how much of what's going on in your body is from nature in the sense like you inherit it from your parents, right? And how much of it is nurture as in like interaction from the environment, right? So the two actually plays equally important role in determining whether people, in this case, get allergies or not, and even getting diseases in general in some cases. So hold that thought because more information is coming in the upcoming slides.
Okay.
Um, yeah. Okay. So we have a problem, right? The problem is we're gonna lose a little bit of DNA every time we copy our DNA, and we don't want to be losing genetic material. That is not very good. Um, so to fix that problem, we have something called telomeres. Telomeres. They are located at the ends of your DNA. Okay. Um, Their job is to protect the ends of the DNA, and they are non-coding DNA. Non-coding means they don't. DNA contains instruction to make proteins, right? I told you that before. So these telomeres, they don't code for anything. They don't make any proteins. They are just like filler DNA, if you would, that are located at the ends of the chromosome. You lose them, no big deal. I don't really care because they don't do anything anyways, other than for the protection. So they are just repeats of TTA GGG. And they literally repeat like, hundreds of times. It's just TTA, GGG, TTA, GGG, like a couple hundred times. And then they're located at the ends. So that's what chromosome looks like, the X's, right? And then the part that lights up at the end, that's where the telomeres are. Okay, like they don't really light up in real life. Everything is just black and white, right? Like in real life, because they're so small. But we can use techniques to cause them to light up. So that shows you where the telomeres are. The best way to visualize this is to think of telomeres as the plastic thing that is at the end of your shoelaces. Okay. If you lose that plastic thing, then your shoelace will eventually fray and break apart, right? So we don't want that for our DNA. We don't want the DNA to unravel. We don't want it to fray. And having the telomeres there is going to protect the ends of the DNA. So essentially, it's like this, right? Think about this. Every time you tie your shoes, like, just imagine if every time you tie your shoes, you lose a little bit of the plastic thing. Okay, so you cut it off, cut it off, cut it off, cut it off. And then eventually, if you tie your shoes enough times, you will lose the entire plastic thing. And then your shoelace is gonna basically break apart, right? And that's what's happening in in cell division. Every time you divide your cell, you need to copy your DNA. And every time you copy your DNA, you lose a little bit at both ends. So your telomeres get shorter and shorter and shorter. Now, it's not a big deal to lose telomeres. As I told you, they don't code for anything. But if the telomeres gets too short, then you shouldn't divide anymore. Because if you divide one more time, you run out of telomeres to cut. You run out of the plastic thing to cut, right? So if you divide again, you will start losing vital DNA. You will start losing important DNA, the coding stuff, the stuff that actually helps you make protein, right? And we don't want that. So when the telomeres gets really, really short, critically short, then the cell is not permitted to divide. Okay, it will send signal and say, you know what, we shouldn't divide anymore. If we go through with another round of division, we're gonna lose important DNA, and it's not good for anybody. So your cells cannot divide indefinitely in the body. Right? It's good for 30 to 40 divisions, and that's it. By then, you will run out of telomeres to lose, and your cell will just not divide anymore. Okay. So here is a question to think about. Right? If you're not already thinking about it, listen to what I have to say. Let's say I was born, and then I got the DNA in all of my cells, and then I grow up, right? I get bigger, I need more cells, so my cells divide. Your cell would divide too. So I started with brand new DNA, and then as I grow up, after every cell division, the DNA gets shorter and shorter and shorter. By the time I'm like 30 years old, my cells probably went through, 20, 30 different divisions already. So my DNA is already short by the time I'm 30, right? Right? So far, so good, right? By the time we are adult, our cells already gone through many rounds of division. So at that point, our DNA are already shortened. Okay. Now, what's the problem of having shortened DNA when you are an adult? Well, let's say, you wanted to have a kid, right? So I had my first child when I was in my 30s. And isn't that a problem? Because my DNA is already shortened. So when I pass the DNA to my kids, won't they start with shortened DNA? And then they when their cells divide, it would get even shorter, and then wouldn't it get shorter for every generation, and eventually the human race would run out of DNA? Do you understand the problem here, guys?
Yes.
Okay, so that's a serious problem, right? Okay, but of course that doesn't happen, and the reason is because of this. I'm gonna skip the this next slide for a second. I'll come back to it. And the reason is because we have something called telomerase, not to be confused with telomeres, okay? We have something called telomerase, which is an enzyme that is found in germ cells. Germ cell, okay? Does anybody know what germ cells are? Nobody was able to tell me this morning. I just want to know if any of you know what that is. In the body, where are germ cells? What are they? No? Okay, that's all right. No worries. Germ cell.
Reproductive cells,
What's that again? Sorry.
like reproductive cells,
Yes,
as in
exactly.
like
Right. So
male
germ
and like
cells,
female.
they are cells that will become sperm or egg. Okay, so for guys, they have germ cells in the testicles. For females, they have germ cells in the ovaries. These are cells that will become the sperm and the egg, and obviously, the sperm and the egg is what makes the baby, right? So, in the germ cells, we have this enzyme called telomerase, and the function of the telomerase is to rebuild the telomeres. So even though, when you're 30 years old, your DNA are short, but no worries, the sperm and the egg that you make are going to have perfectly long DNA because of telomerase. It rebuilds the telomeres. Okay, so that you have brand new plastic caps at the end, and your kids are going to start with brand new long DNA. So without the telomerase, it gets shorter and shorter and shorter, and eventually you're not allowed to divide anymore. But with telomerase, it stays long, and the cell can reset itself and start have a new beginning, if you would. Okay. Let's go back to Dolly for a second. I don't know if many of you have heard about Dolly. It was the first mammal to be cloned from an adult somatic cell. A somatic cell means like a body cell. So anything that's not germ cell is called somatic cell. All right. Your liver, your muscle cells, those are somatic cells. So it's the first mammal to be cloned from a somatic cell using something called nuclear transfer, which I'll explain in a bit. This happened in 1996, long time ago, right? And the lifespan for a normal sheep is about like 12, 13 years. But for Dolly, she only lived for 6 years. Okay, so I'm going to explain to you why that is the case. I think it's really interesting and it really connects to what we are talking about. So please try to understand. Let me ask you a question, see what you think. I already told you Dolly was the first mammal cloned, right? First mammal to be cloned. How many sheeps do you think it took to clone Dolly? I'm not talking about like the failed experiments, right? I'm talking about like the successful one that ends up creating the Dolly. How many sheep needs to be involved to clone the sheep? What do you think? Just give me some wild guesses, or maybe you know the answer, right? Just type them in the chat box. It's
Two,
interesting.
male
Okay,
and female.
two, male and female. Okay, great. Anyone else? Two. Okay, also two. Anybody think it's not two? Two. Okay, a lot of people say two, right? By the way, do you guys know what cloning means? Like, just let me just clarify so that we're not confused. Like, if you were to clone me, it means you're gonna create an exact copy of me, right? Okay. But when in this hypothetical world, when my clone is born, it's not gonna be born exactly like me, like now, right? It's gonna be the baby version of me, right? And then it's gonna grow up to be like me. Do you understand that? Okay, so that's what we're talking about, right? So how many sheep did it take to clone Dolly? Okay, well, I'm gonna tell you right now. So this is the first sheep. Okay, this is the first sheep. This is called the nuclear donor. Right? So we're gonna take a somatic cell from this nuclear donor. Okay, and it was a female sheep. And this would be the cell. A somatic cell has a full set of genetic information, right? Like all the genetic information of the sheep. And then what they did is they remove the nucleus. Okay, so there is the nucleus. You know nucleus, right? From lecture 3. It contains all the genetic information. Then we have another sheep here. Okay, also a female sheep. And what they did was they take an egg. Okay, they take an egg. Just gotta draw the egg in red. Now, egg only has half the genetic material. Think about it, right? We have 46 chromosomes. Eggs has 23. Sperm has 23. Sperm meets the egg. That's 23 plus 23. The baby has 46, right? So eggs and sperm only have half the genetic material. And what they did here is they take the eggshell, so to speak, shell. Okay. When I say egg, right, I'm talking about like sperm and egg, right? Because someone was asking me, they were like, I didn't know sheep lay eggs. No, they don't lay eggs. Okay, I'm not talking about like chicken eggs or like duck eggs, right? Like I'm talking about like sperm and egg. Okay, so you take the egg and then you just keep the shell. Okay, you just keep the shell. You remove the nucleus, and then you put these things together. Okay, you put these together. You literally just inject the nucleus from the nuclear donor into the shell of the egg donor. So in the end, what do you get? You get an empty shell with the full set of chromosome from the nuclear donor. Okay, then we need a third sheep. Okay, surprise. We need a third sheep where we put the egg in it. What do we call a person who carries baby for another person? Because that person might not be able to have their own kids.
Surrogate. Very good.
That's right, the surrogate. This is the surrogate mother, okay? The surrogate sheep, if you would, okay? And it provides the womb for this egg to be implanted, okay? Implant into surrogate. Okay, and then the baby grows, and then you end up having Dolly. Dolly. Now I want to ask you, in this process, Dolly is a clone of which sheep? Is it a clone of the nuclear donor, clone of the egg donor, clone of the surrogate sheep, or really is a combination of all of them? What do you guys think? Let me know. Type it in the chat. Unmute.
Clone of the nuclear
Combination.
donor.
The nuclear donor, yeah. Anyone else?
Combination.
Combination, okay. Anyone else? Okay, Blessing also say combination. Justine say one. What was the other one? Lemio say two. Well, very good that you guys have different opinion. But the correct answer is Dolly is clone of number one. It's gonna grow up to be exactly like this sheep, right? Genetically is the same. Why? Why? Because that's the one that gave all the genetic information. Look, you took the nucleus from this sheep, and the nucleus is where you find the DNA, right? This egg donor over here, only gave the eggshell with no genetic information in it. They removed the nucleus from the egg and replace it with the nucleus from the nuclear donor. So all the genetic information is identical to the nuclear donor. The surrogate also provides zero genetic information. It only provided the womb, right? Okay. I mean, imagine this in the real world. The surrogate mother, like in a real world scenario, is also just providing the womb, right? It doesn't give any genetic material to the baby either, right? So that's why Dolly is only the clone of number one. Does that make sense? Yeah.
When you're cloning, so the male sperm.
Yeah, there's no sperm in this case, right?
Sperm.
There's no sperm because
Okay.
you remove the egg nucleus, okay, and then you put a cell nucleus in it, which already has the full set of chromosomes, right? You didn't need to, because if you fertilize it with a sperm, then you are not cloning. You're actually just, doing in vitro fertilization, right? Cloning means exactly the same genetic material as the donor. All right. And Blessing, you had another question?
Yes. Without the egg, how would Dolly be born?
Without the egg, like
Yes.
this egg over here?
Yes.
No, well, no, you need the egg, right? But not the genetic material from the egg, but just the cytoplasm and all the nutrients in the egg, right? So you need the shell of the egg to put the nucleus in. You understand
it's just
that?
the shell, not the egg itself.
The whole thing is the egg, but after you remove the nucleus, it's just the, like when I say shell, I mean like, the cytoplasm along with the mitochondria and all that stuff, right?
Okay.
Does that make sense?
Mm-hmm.
Okay. Now, here is the interesting part, right? The whole story kind of builds up to this point. Dolly was born, right, in, like I said, July 1996, right? So, when it was born, how old would we say Dolly was? Well, when someone's born, we say they are newborn and they are like supposed to be age 0, right? So to speak, right? Age 0. However, because Dolly got all the genetic material from the nucleus of a donor sheep that is already an adult. So Dolly, even though it was born age 0, biologically, it's already old. Biologically, it's the same age as the nuclear donor, right? The nuclear donor, up to that point in its life, has went through many rounds of division. So the genetic material, the DNA is shortened, right? We are not using any germ cells here. So we didn't reset the telomeres. Whatever length it was at that point of the life, that's the length that get passed on to Dolly. So again, even though Dolly was born as a baby, but on the genetic level, on the cellular level, is already quite old. And that's why it only had half the lifespan as a normal sheep. Do you understand that? That's why we can't just like go around and cloning people. Like, yeah, exactly. It looks like a baby, but genetically, it is old, and that's why it's not going to live very long, right? It's like it's born old in a baby shell, if you would. Like, does that make any sense?
So this means that if the nuclear donor was like a year old, Dolly would have lived longer.
Exactly, exactly. Very good. Okay, because it copied the genetic age of the donor, right? So if I don't know if I don't think they repeated this experiment, but you know if they did and then they use like a younger sheep, probably Dolly would be like Dolly 2.0 would be able to live longer. Does that make sense?
Yeah.
Yeah. Okay. I hope the rest of you are still with me and you know as excited as I am about this stuff because it's really cool. It's really interesting. Of course, like cloning is you know, not there's obviously there's a lot of ethical issue that surrounds it, uh, but it's not doesn't always have to clone the whole person, right? Like if we are able to clone organs, right, then it would eliminate the problem of organ shortage, right? You know, do you guys know what's the number one the longest wait list when it comes to transplantation? What kind of organ is the longest wait list? Does anybody know?
Heart?
Uh, Good try, not the heart. You have two of these
Kidneys?
things in the body. Exactly, kidneys. That's the most weighted, like the longest wait list in Ontario, and you know I think in other country as well. Like people just can't get enough kidneys, right? Because of mismatch and other problems. Liver is also up there on the list, but liver is a little bit better because did you know if you cut 1/3 of your liver and donate it to someone else, you can grow your liver back? Like in within a year, you will get your full liver back. Liver is amazing in terms of its regenerative capability, but you cannot like grow another kidney back, right? Like that's just not gonna happen. So there you go. If we are able to like you know clone our own organs, then that's gonna be a good thing. Abnet asked why telomerase enzyme is not working for Dolly. Okay, great question. Telomerase is only turned on in germ cells when you make sperm and egg, right? And since they did not create Dolly using sperm or egg. Sure, there was an egg involved, but then they didn't use the genetic material from the egg. Remember, they took the nucleus away, right? All the genetic material came from an adult cell of the donor here, right? So there is no telomerase involved there. Right? Whatever the length was at the point of cloning, that's the length that Dolly's gonna get. All right. Now, let's move on to the next part here. Some people like to, sometimes they ask me, right, in class, they're like, so if telomerase can reset the telomere length, wouldn't it be a good thing to have telomerase like in all of our cells so that the DNA don't get short and you know our cells can divide for longer period of time and maybe we can live longer? Like it sounds like the limitation here is how long the DNA are, right? So if we were able to turn on telomerase, wouldn't we be able to live longer maybe perhaps or like we don't age as much? Well, it turns out telomerase, when turned on in regular cells, so other than germ cells, if we turn on telomerase in somatic cell, that is a problem. That can turn regular cells into cancerous cell because cancer basically is uncontrolled cell division. Okay, and one of the things that limits the division is the shortening. Okay, so after a cell divide, again, like I said, 30 to 40 times, the telomeres will be critically short and it's not allowed to divide anymore. But if you turn on the telomerase, then the cell all of a sudden can keep on dividing and dividing and dividing. And that could cause a tumor to form. And the tumor can be benign, which is a good thing. It's not cancerous. You can surgically remove it, and most people are fine. But if it's malignant, then it is going to be cancerous. Okay. So that is actually what happens when people look at telomerase in cancer cells. They found that telomerase is often elevated. There's a lot of telomerase in it. Right? You wouldn't expect it to find it there, but there is a lot of it, and that caused the cancer cells to become immortal. Essentially, remember the HeLa cells that we were talking about earlier in the course? That's essentially immortalized cell. They can keep on dividing forever, partly because of the telomerase. So one of the hot area of research nowadays is to look at telomerase inhibition as a potential treatment for cancer. Right? If you can block the telomerase in these fast-growing cancer cells, maybe you can stop them from dividing. If you stop them from dividing, maybe you can shrink the tumor and improve the prognosis, the outcome of the disease.
Sir,
Yes.
is that how cancer spreads, like when the cells divide?
Spreading is another thing, which we will look at as well. The end of this lecture, we won't get to it today, but the end of lecture 6, we will have a dedicated section for cancer. We will look at the characteristics of cancer, and one of the characteristics is the ability to metastasize. You probably have heard of that word, right? That's like spreading to different parts of the body.
Yeah.
Okay. I don't know. Do you guys feel like doing polls? I'll do one. I'll see if you guys like participate. If not, I'll just give you the answers. What? Actually, I'll just do it myself because we don't have too much time today. What is a key difference between DNA replication in prokaryotes and eukaryotes? Okay, so let's see. Prokaryotes have telomeres and eukaryotes do not. That is not true. That is actually the other way around, right? We are the one that needs the telomeres because our DNA gets shorter and shorter. Prokaryotes don't have this problem because they have circular DNA. They are able to just, copy in a loop, and it's not going to get shorter. So they don't need telomeres. Prokaryotic chromosomes have a single origin of replication, whereas eukaryotic chromosomes have many. That's true, right? I told you ours is very long, so it only makes sense to have multiple bubbles to speed up the process. Prokaryotes produce Okazaki fragments, but eukaryotes don't. That's not true. We both do. Okay, both of them have Okazaki fragments, and both of prokaryotes and eukaryotes have RNA primers. So the answer is B. Okazaki fragments are joined together by ligase. Okay, ligase. To ligate is to join two things together, to glue them together. So we need the ligase. RNA primers are removed by, which one is this, guys?
DNA polymerase 1?
That's right. That's the number 1. Right? Number 3 is going to fall off. It's going to be like, hey, number 1, come over here. Please remove the primer. Right? That's what it does. Very good. Telomerase are needed to, let's see, re-lengthen shortened chromosomes in some cells. That's correct. Okay, and that some cells is like germ cells, right? Germ cells, and actually some stem cells in the body as well. And stem cells are cells that can become any cells in the body. We have stem cells in the blood. We have stem cells in the gut. Okay. Prevents cancer development? Nope. That actually makes it more likely to have cancer if you turn it on in regular cell. I'm talking about. Remove non-coding repeats? No, it adds. Right, that's how it re-lengthens it. It adds more of the repeats. It lengthens the ends of chromosome. So the answer is just A. All right, that's all. Perfect timing. When we come back in 5 minutes, we will start the second half of the lecture, which talks about the cell cycle and the actual cell division. All right, see you guys in a bit. 5 minutes. Okay, let's keep going. For the first half of this lecture, we talked about how the DNA is copied, and the process of copying DNA is an important one that precedes the actual division. We can't divide, we can't split the cell unless we have copied all the genetic material, right? Otherwise, your cell, whatever you get in the end, is going to be missing some genetic material. So, DNA replication has to happen, excuse me, before the actual division. So now we're going to look at the cell cycle. One of the parts of the cell theory states that all living things, all living cells come from division of pre-existing cells. Okay, in other words, to get more cells, you need to take an existing cell and cause it to divide. Okay, well, nobody really fully know where the first cell came from. I mean, there are theories about it, but no one knows for sure. But we know if we need to get new cells, we have to divide the existing cells. The purpose, why do we have to, do divisions at all? Well, the most obvious reason is replace lost or damaged cells. If you fall down and then you have a cut, you lost some cells, you damaged some cells, you need to replace them. So we need to divide, use the existing cells, tell them to divide, and we get new ones to replace them. Growth and development, one of the characteristics of living things, right? Um, we have a lot more cells now than we did when we were a baby. We are bigger, so we have more cells. So all those extra cells has to come from somewhere, and that would be, again, the purpose of cell division. And we, not humans, but in some organisms, the cell division is important for reproduction. Okay, in what we call like asexual reproduction. Okay? Humans and many animals, we do sexual reproduction. Sexual reproduction means it requires two individuals, each contributing half the genetic material. So like the father contributes half through the sperm, and the mother contributes the other half through the egg, and then you end up having the baby, and that creates genetic variations, right? That's why our kids look different from each other and from us, which is probably a good thing. But for some organisms, they solely rely on asexual reproduction, which is just the organism by itself, and they don't need a partner, and no fertilization between eggs and sperms are necessary, and they are able to just make copies of themselves. They essentially clone themselves. Okay, so the advantage is you don't need to find a partner. It's hard to find a partner in like say in the ocean, right? Like how do you even bump into another like I don't know, like whales or something. Like it takes time, right? So like for some organism, it's really hard to find a partner. So it makes sense to do this kind of reproduction. It's fast and it requires less energy. So did you know if you take a starfish, sea star starfish, and you break off one of the arms or something, uh, that arm can grow into another starfish. So you end up have two starfish that are like clones of each other. That's how they reproduce. Amoeba, a lot of these, single cell organism that we talked about that lives in ponds, they're able to, perfectly happy to clone themselves. No partner is needed. Plants, a lot of plants do this. If you grow any plants at home, like sometimes when your plants get too big, you can cut off a piece of it, just leave it in water, and then it will start to root and then you can put it in a new pot, and then you get another plant, but that plant is genetically identical to the one that you got the piece from, right? Okay. I saw something over the weekend. They're saying this never occurred to me, but next time you eat a banana, do you realize there's no seed in the banana? So how do they grow bananas if the bananas is seedless, right? So apparently they grow them by cloning them, much like you clone your plants. Like you don't need to pollinate the plants, right? You just take a piece off and then let it root and then it will grow. So that's kind of like what they do with the banana plants. They take off like a shoot from the actual banana tree, and then it's able to grow into a new tree. So the problem with that is all the trees in the plantation is going to be genetically identical. So if there is some kind of disease that targets that hurts for the tree, one of the trees, all the trees are likely to be susceptible to it. All the trees are likely to be hurt by it, right? Take humans for example. By now you probably either have caught COVID or know someone who got COVID. Um, I actually got it like last week during the week off. I'm fine now. But some people are more affected by it than others, right? Some people get really sick, others don't, and they think there's a genetic component to it, right? Okay. So if all the banana trees are clones of each other and they are genetically identical, when a fungus comes, that causes one tree to basically die off, they are all likely to get wiped off because they are all the same. And that's what's happening. There is such fungus that exists. It's harmless to human, but then they really kill the banana trees. And if they don't find a cure soon, the program I was watching, they're saying, the bananas are going to be very expensive soon because they won't be able to harvest as much anymore. So that's the biggest disadvantage of cloning, like low genetic diversity. Okay, it's you're not like mixing the genes like we do in sexual reproduction. Okay, so just a quick recap on the structure of DNA. As the naked molecule on its own is the double helix with with the sugar phosphate backbone and then the nitrogen base and all that stuff. But it's really long and strainy, and we can't pack that into the nucleus. And that's why we need to wrap them around proteins to form the chromatin, right? You may remember that is the state of DNA that exists inside the nucleus. But when you have to move the genetic material around, like when you are trying to uh, separate the DNA when the cell divides, then you need to condense it even further. And the most condensed form is called chromosome. Okay, so chromatin and chromosome are essentially the same thing. Uh, it's just that one form is more condensed, more packed compared to the others. So right now, if your cell is not dividing, the DNA exists as chromatin. It only becomes chromosome when you need to move them around, okay? Now we have 23 pairs of chromosome. You should know that by now. I have been like dropping this number every now and then in the course. So 23 from each parent, and we have 46 in total. Of the 46 chromosome, 22 pairs, 44 of them, are autosomal chromosomes. Okay, 44 of the 46 are autosomal chromosomes. 22 pair is 44, right? Okay, so we can call them autosomal chromosomes or autosomes. They determine everything about you except for the sex. So things like eye color, skin color, to a certain extent, your height, whether your hairline is like some people have hairline that looks like this. We call that widow peak. Some people have straight hairline. Yeah. Curly hair, straight hair, whether your earlobe is attached, detached, your blood type, all this stuff, everything about you is written in the autosomes, in the 22 pairs of chromosomes. The 23rd pair is the sex chromosome. If you are female, then you have two X's. XX is female. Male is XY. All right, only male have Y chromosomes. So I want to ask you, who determines the sex of the baby? The father or the mother?
The father.
The father. Anyone else?
The father.
The father, that's right, because the father is the one that has the Y chromosome, right? So there's a 50% chance that your the father is going to be able to pass on the Y. If the sperm carries the Y and fertilizes the egg, you get a boy. If it's the sperm that's carrying the eggs that fertilizes the egg, then you get a girl. Okay, so it's 50-50. And the event is independent. That means even if you have 3 daughters already, what's the chance that you're gonna have a son the next time? Still 50%. Okay, what happened previously is not gonna change the odds of future outcomes. Okay, like you flip a coin 4 times and it's heads. Right, next time you flip the coin, it's still 50-50. You understand that? So it's just a fun story. My grandmother on my mother's side, she is like super traditional. Okay, she's like in her 90s, right? And I have 2 daughters. I think I told you multiple times already, right? So like she's not very happy about that. Okay, sometimes she would, when it was just me and her sitting down, she would bring this up and she's like, what's wrong with your wife? And I was like, what's wrong with her? And then she's like, why is she only giving you daughters? She should be giving you son. And then I try to explain to her, like, grandma, it's actually my fault. Because I am the one who, decides whether is boy or girl. It has nothing to do with my wife. But she just wouldn't hear about it. She's like, no, it's the wife. It's always the woman that decides. Right? And she's like, I would know because I have 3 sons. So that's very very set in her mind, right? About this thing. But now you know. If you didn't know, now you know. It's the male. It's the male that determines the sex of the baby because of the Y chromosome. Okay, when it comes to chromosome size and numbers, it actually has nothing to do with how big the organism is. Okay, humans has 46, as you know, but like goldfish, which is much smaller, they have 100 chromosomes in each cell. Okay, something like a snail, which is even smaller, has like 54. And so there's no relationship between the number of chromosomes and the kind of organism. It seems to be a little bit, arbitrary, a little bit random. Nobody really knows why. All right, the cell cycle. If you look at the lifespan of a single cell, okay, it would be spending most of its time in what we call the interphase. Okay? On average, we would say that the cells spend like 90% of the time in interphase, just performing its normal function, growing, preparing for the division. Right? The division is eventually going to happen, so you gotta prepare for it. You gotta gather resources. You gotta check your DNA and all that stuff, and all that happens in the interphase. Right? 90% of the time, the cell will spend in this interphase. Of course, there are variations, right? Some cells divide more rapidly than others. Your skin cell, not the one on the surface. The one on the surface are all dead, by the way, in case you didn't know. But it's the one that it's in the deeper layer, the one that gives you new skin, right? Those divide quite quickly. Okay, like every couple of weeks you replace all your skins, right? That's why we shed like dead skin cells all over the place. But some cells never divide. Do you have any examples where the cells don't divide? Like you lose them, it's gone, they don't come back. You must have heard people say that before, right? Give me some examples. Brain cells, right? Brain cells. You lose brain cells, they don't come back. That's why people who suffer from stroke and some brain cells die, they are permanently affected by it, right? You can't get new ones coming back. Same thing for heart cells. Okay, so same thing for heart cells. That's why you can never heard of you have never heard of heart cancer. The heart cannot get cancer. The cells don't divide. If the cells don't divide, you can't get cancer. Okay, now I know what you're thinking. You know, I've heard of brain cancer, but you just told us brain cells don't divide, right? Turns out, you will learn this from me next semester. There are two kinds of brain cells. There are those that don't divide. Those are the one that helps you do the thinking and all the stuff. And then there are brain cells that protects the neurons. Those are the ones that can give rise to cancer. Okay, but generally, like if they can't divide, then they will be stuck in interphase forever, right? Yeah, they don't replace themselves. So mitosis is the actual cell division. It's the part where it actually split into two daughter cells. And you know, if the cell's gonna do it, it's not gonna spend too much time in it. She's gonna be doing it like 10% of the time, let's say. So this whole purple part here, that's the interphase. And inside interphase, actually is separated into three parts. Okay, there's G1 and G2, like getting your driver's license in Ontario, and then there's the S phase. Okay, so very briefly, we're gonna come back to this in a bit, but G1, this is when the cell asks, you know, are we ready? Are we ready to divide? Okay, so there is a like a checklist to go through, right? For example, uh, is the cell big enough to divide? Like, I mean, if it's very small, we might not have enough cytoplasm to split, right? So is it big enough? Do we have enough nutrients to follow through with the division? How about the quality of the DNA? Right? Is there any damage? Do we need to repair it? What about the telomeres? Are the telomeres long enough, or are they very short already and we cannot go through with another round? Right? So it asks all these questions. It does all these checks and see if the cell is good to go ahead. Sometimes it's not. Sometimes, you know, the cell is too old, there's too much DNA damage, then it might kill the cell, right? Apoptosis. We learned about that. The cell might stay the way it is and not divide. But if all is well, everything is green light, then we are going to move on to the S phase. S is for synthesis. This is where you are going to be copying your DNA. It's the stuff that we learned in the first half of the lecture 6. Okay, you're gonna be opening the bubble. You're gonna be copying all the DNA. And then afterwards, you're gonna do another check, right? Okay. Did everything copied? Okay. Like, did you make any mistake when you do the copying? Did you copy all of the genetic material? Do we need to, you know, do any kind of repairing and so on and so forth? And again, if it doesn't pass the G2, then it's not allowed to move on to the next part. So if everything is okay though, it will move on to the mitosis, the M phase, okay, where you will be able to, you know, actually divide the cell. Any question?
Yes.
Mhm.
Let's say if one person has one chromosome less.
One chromosome less, so like
Chromosome,
45 instead of 46?
yeah,
Mm-hmm.
that's normal in some cases,
Okay, so interesting
right?
point you bring up. Yeah, you know, every time you ask a question, I keep on answering and tell you that we will talk about it later, but we actually
Okay.
will. So the short answer, people with missing chromosome usually will not survive as the embryo. Okay, so these leads to like natural miscarriages. But people with extra chromosome are the ones that tend to survive, and they often will have some kind of conditions. Right? For example, Down syndrome, that's extra chromosome 21, right? Okay.
Mhm.
And depends which chromosome is extra. And sometimes you don't even have the whole chromosome as extra. You just have bits and pieces of chromosomes that are extra. Those tend to lead to, you know, leukemia, for example. It's very common to have extra chromosomes. A lot of cancers have extra chromosomes, bits and pieces here and there. Okay. So missing
Okay.
chromosome, usually not viable. extra chromosomes, quite common in the population.
Okay.
Yeah. So let's take a closer look at the interphase. Like I said, this is the phase where the cell spends most of its life in, just performing its normal function. If it's a muscle cell, it'd be contracting. If it's a liver cell, it's going to be maybe detoxifying your blood. Whatever the function, it's just going to be doing it in the interphase. Near the end of this phase, the cell will prepare to undergo mitosis. It's going to go through the G1 checkpoint, and then it's going to duplicate the DNA if everything's good, and then it's going to have another checkpoint afterwards. So in this picture, we can see three different colors. And again, they are not really like actual colors of the cell, right? We just use a staining technique to cause them to be different colors. We have the blue that shows you where the DNA are. And right now I can't see anything here. Okay, like I mean, I see the nucleus, but I can't see the DNA on the inside because they are still in the form of chromatin. Okay, it's very loose. You can't really see it if it's not like compact, right? And then we have these spindle fibers. You might remember from lecture 3. They are like a type of cytoskeleton that helps guide the movements of DNA. And then we have the red that just kind of shows the cytoplasm. Okay. All right. So here we have 2 chromosomes. Right? Like I said, we have 23 pairs. And the chromosomes are grouped together based on their lengths. So chromosome 1 will have the same length as chromosome 1, and then 2 will be shorter than 1, and then 3 will be shorter, and so on and so forth. But number 3 will be same length as number 3. Number 5 will be same length as number 5. And one of it would be from each of your parents, right? So let's say this is chromosome 4 from mom, chromosome 4 from like your mom, and then in your body, right? Chromosome 4 from your dad, and then they will be the same length. Now, do they contain the exact same information? No, because like your parents don't look like each other, right? Like some features might be the same. Like for example, one might have blue eyes and one might have brown eyes. But other features might not be the same. Sorry, other features could be the same, right? Like for example, they could have the same skin color, right? They might have the same blood type. So things could be the same, but they are not necessarily the same. Okay? So on chromosome 4 here and here, they would have the same type of information, but not necessarily the same information. What do I mean by that? Like at this location, it might be about eye color. So you would expect to have eye color on the other side as well. But they might not be the same eye color, right? Like over here, it might be blood type. Then you would also expect to find blood type on the other one. But again, they might or might not be the same blood type. Okay, so it's the same series of information, but the version of the information might be a little bit different. Okay. So after DNA replication, you would copy each one of them, and they are tied together at this place called the centromere. Okay? And these chromosomes that are attached to each other, we call them sister chromatids, TID. Don't confuse with chromatin, TIN. That's the DNA with the proteins, and this is chromatids, which means they are connected together. They are exactly the same. Now, this is a little bit weird. So I want you to go through this with me. Okay, tell me, how many chromosomes do you see here? It's not a trick question. Just tell me, how many chromosomes do you see in here? Hopefully, you will tell me the right answer.
2.
2, right, exactly, right? I hope the rest of you also say 2. Okay, 1, 2. And then it gets tricky. How many chromosomes do you see here? Okay, what do you guys say? I want you to do this with me so you don't get it wrong on the test.
4.
4, yeah. 4, say, Matrice also say 4. Anybody think it's not 4? You should voice out if you don't think it's 4. Okay, Well, most people think it's 4. But the way that the scientists decided to count chromosome is this: if they are stuck together, you still call it 1. And so even though like you see 2 strands here, but because they're stuck together, we call this 1. And so this is 1 and 2. So even though you copy them, afterwards we still count them as 2 chromosomes, even though there are like 4 chromatids. All right. So please keep that in mind. You know, as we continue to discuss what happens in mitosis. Okay, so there are 4 phases in mitosis: prophase, metaphase, anaphase, and telophase. You can remember like PMAT. And that's the order that it happens in: prophase, metaphase, anaphase, and telophase. All right, let's take a look at prophase. Right? We're gonna look at the first part, right? Prophase, the early prophase and like late prophase. At this point, DNA replication is completed. That happened in the interphase, right? Okay, so that already is done. If the cell is allowed to go to prophase, it means quality of DNA is good. Cell is ready to split. So at this point, the chromatin is going to pack even further to form the chromosomes. Okay, we're going to be moving the DNA soon, so we got to pack them up so that they're more compact. It's easier to move them around. So if you look at the nucleus, you can start to see the outline of the DNA, right? Whereas before it was just like a sea of blue with nothing distinctive. Like I said, the DNA replication is completed. The sister chromatids, essentially the copies of the DNA, are held together at the centromere. Just go back here to show you this, the place where you tie them together, that's the centromere. We gotta get the chromosomes out of the nucleus, so that's why we need to remove the nuclear envelope. And then you might remember the centriole, right? The centriole is the organelle that sends out the spindle fibers. If you take a look at the middle of this picture here, you can see the 2 yellow dots in the center. Those are the centrioles, and then all these green fibers that are like coming out, those are the spindle fibers. Okay, remember the purpose of the spindle fibers is to help move the chromosomes around. As prophase continues, the chromosomes becomes fully formed. You can see the individual outline of the chromosome now. It's very obvious, right? You can make out the individual pieces. And the nuclear envelope is gone, right? That's why all the chromosomes are like outside now. And the spindle fibers will attach to the centromere of the chromosome. Okay, so P for prepare. Okay, we are preparing to split the cell in prophase. We got to get everything ready. We take the chromosome out, send out the spindle fibers. We are ready to move. So remember PMAT, after P is metaphase. M is for middle. Okay, so in metaphase, the spindle fibers is gonna have a tug of war. It will pull the chromosome back and forth until they all line up in the middle of the cell. Okay, that's all. Hey, how many chromosomes in this picture, guys?
4.
4 is correct. 1, 2, 3, 4. Excellent. Anaphase. A for apart. Okay, so after the tug of war lines up the chromosome in the middle, they will pull and split the sister chromatids. Okay, so the X that we had before? Remember, like each half is actually genetically identical, right? They're copies of each other. So when you split it, you half goes each way. So how many chromosomes here?
8.
8 is correct, right? So if you have 4 in metaphase, it's gonna double in anaphase because now you're splitting them, right? So in anaphase, you pull them apart. And then finally, in telophase, T for tearing apart. Tearing apart. We are basically, like if you think of like one giant ball of Play-Doh or dough, right? You just split in the middle, now you get 2 circles, you get 2 cells. Okay? Everything is backwards now compared to prophase. We done moving the chromosomes, so let's turn them back into chromatins. We need to form the nuclear envelope again so that we get our nucleus back. We no longer need the spindle fibers. We're done moving things around. We don't need to move them anymore. No need for spindle fibers. And then we're going to split the cell in a process called cytokinesis. Okay, that's just a fancy word for dividing the cytoplasm. Right? Each gets roughly half and half. All right? And that's it. So this is onion cells, the root tip of onion cells. And we want to identify the phase of mitosis that we're looking at. Okay? What do you think this one is? This is a cell. Help me out. You think this is prophase, metaphase, anaphase, telophase? What's going on? That is good, metaphase. Very good, because it's all in the middle, right? It's all in the middle. How about this one? Anaphase, very good. You can see that they are being pulled apart. Okay, how about this guy? Okay, we have prophase. Anyone else? Yeah. This one is cytokinesis or telophase. You can see the 2 nuclei are forming, right? If you look really closely, you can see a line is starting to form in the middle. Right? So this is telophase. Okay. And over here, this would be prophase. Got it? So on the quiz, I think that's gonna be in like 2 weeks or something. Like you might see a picture, and then I'll ask you, what are you looking at?
What's B? Is it
What? Which one?
B? B?
This one? This one?
Oh,
Oh,
that's
B is
B.
talking about, yeah, not this one.
Oh, okay.
But yeah, sure. Like this here, that's just interface, right? Like it's not doing anything.
So yeah.
Like
Okay.
I can't even see the chromosome because they are in chromatin form, right? Makes sense?
Yeah.
Right, so
Yes.
in fact, like there's a whole row here. Like they are all in interface. They're not doing anything. All right, moving on. So the cell cycle has the checkpoints I told you, right? The G1 and the G2, and essentially they are safeguarding that controls whether or not the cell cycle can progress. Okay, if there are some errors, if there's some mistake, you are not allowed to divide. All right. Now we are running out of time a little bit, so I'm gonna skip these concept check questions. I'll give you the answer. The answer is A. You just write it down and then you do it later, okay? The answer is A here, and then this is C. Okay, A and C. Just write down for now, and then you can check them later. We talk about this later. No time. Okay. One last thing that we're gonna look at today is take a closer look at the checkpoints. Okay. So hopefully I have instill in your mind what the checkpoints are. The checkpoints control how fast the cell is allowed to divide. And essentially, there are 2 kinds of signals here. Okay. Um, First of all, the checkpoint. What's doing the checking? what's checking the DNA quality, what's checking the quality of the, the cell and all that stuff. They are actually proteins that are doing the checking. Okay. And proteins are, of course, encoded by DNA. And so it's possible to make mistakes, right? If there's a mutation in DNA, the protein that you end up making is potentially going to have problem. So we're going to look at that in a second. But we have two kinds of proteins here. We have growth factor, proteins that serve as growth factors in the checkpoints, in the G1 and the G2. And their job is to promote mitosis. Right? So you can think of them as green lights. Okay? Green lights light up, you go, you divide. Okay? On the other hand, we have growth inhibitors, which are like red lights. They stop, they inhibit mitosis. Okay? You're not allowed to divide. Maybe there's some DNA damage. Maybe the cell is too old. A bunch of other reasons. Got it? Growth factors, go and divide. Growth inhibitor, don't divide. So we need a balance between the two. You can't just let every cell go divide, right? That's going to take up too much resources. It might even turn cancerous. We can't just stop all the divisions. Then we won't grow and we won't heal. Terrible thing, right? Need the right balance. All right. These checkpoint proteins are encoded by DNA. And I don't know if I told you this before, but a gene is basically a DNA segment. It's a chunk of DNA that gives the instruction to make a particular protein. Okay? So in this case, we have a gene called proto-oncogene. Proto-oncogene. The proto-oncogene codes for growth promoters. In other words, they have the instruction to make the proteins that acts as green lights. Okay, so they help the cell grow. They help the cell divide. Okay, so we call that a proto-oncogene. And because genes are DNA and DNA can change, okay, when it mutates, when it changes, or sometimes if you have too many copies of it, then the proto-oncogene becomes a bad gene. And this bad gene is permanently turned on or permanently activated. Think about the green lights. The proto-oncogene is the green lights essentially. If you have a green light, that is always green. It doesn't turn off because it's malfunctioning. It's always green light. What's going to happen? Well, the cell will think it's able to divide all the time, right? Hey, nothing's stopping us. It's green light all the way. Let's divide. Then the cell gets out of control and then the person ends up having a tumor. And tumor, again, could be benign. That's the best case, right? It's not harmful. But it can also be malignant, which means is cancerous. Okay? The bad gene is now called an oncogene. Oncogene. That's why people with cancer, they have to see an oncologist, right? It's the same kind of prefix here. Okay? So proto-oncogenes, that's the normal version. Everybody has that. Okay? It helps our cell grows and divide, and we need that. But when it turns bad, then it becomes an oncogene. Then it's always green light 24/7. Then the cell will keep on dividing and does not know how to stop. Any questions? Okay. The opposite of proto-oncogene is tumor suppressor genes. These are genes that encodes for the growth inhibitors, the red lights. Okay, so they can be things that slow down cell division. They could play a role in repairing DNA mistakes, or they tell a cell to go have apoptosis, kill them off, right? When the tumor suppressor gene does not work, then that's like not being able to turn on the red lights. Okay, so without the red light, it's the same problem, right? They won't stop. It will just keep on dividing and dividing. And again, that could lead to cancer. So the kind of mutations that we have in proto-oncogene is called gain of function mutation. Gain of function mutation means that it's permanently switched on. That's basically what it means. Okay, permanently turned on. So if you think of a car, how do you make a car go faster? Well, you step on the gas pedal, right? And in this case of a gain of function mutation, it's like having a stuck gas pedal, okay? And it will keep on accelerating, right? It won't stop. So only one mutation is enough for this to happen. So you already know we have two copies of each gene, right? Because we inherit one from each parent. So if you think about the green light situation, it's like having two green lights. Okay, two green lights. One green light here, and then a second green light. Each from one of the chromosomes that you have, right? So imagine in a normal situation, the green lights would turn on, and that means the cell can divide, and then the green lights would turn off when you don't want it to divide, right? So it's on sometimes and then off sometimes. However, with the gain of function mutation, right, it's permanently on. One of them is permanently on, and that's enough to cause trouble, right? Because if one of them is always on, it doesn't matter the other one is functioning. Your cell is not looking at the other one. It's looking at this one that's like always green. It's like, hey, let's go, let's divide, right? Let's not stop. And so that's why only one mutation is enough to cause cancer for proto-oncogene. Okay. On the other hand, the tumor suppressor gene is red light. Okay, so think of the red light. So again, the red light, normally speaking, it would turn on and then it would turn off. When it's on, it's telling the cell to not divide. In this case, it's a loss of function mutation. Okay, loss of function means it means it won't turn on. The other one is gain of function. It's always on, right? So here, let's say, again, normally they would turn on and then you would stop the cell from dividing. But let's say you have a mutation and then it causes one of them to not turn on. That's okay. I still have the other lights, right? When the other light turns on and say red, say stop, don't move, then the cell will not divide, right? So even if you have one that's not working, even if you have one mutated copy, it's okay. It's not cancer yet because you still have one backup. You still have the other light that's functioning. It's only when both of the lights are broken, then there is no way to stop the cell. Okay, think about driving again. Like if your brakes are not working, I guess you can still kind of slow the car down with the hand brakes, right? Like there's like two different ways to stop the car, right? It's not like the stuck accelerator, like one problem, then the whole car is out of control, right? So for this reason, people who are born with only one functional copy are more likely to get cancer, right? So people might inherit one bad copy. So in all of their cells, only one red light is working. So if there is a mutation that happens in any of the cell, that's enough to cause cancer. But for people who have regular genes that are not mutated, it would take two mutations in the same cell in order for the cancer to happen, right? So for that reason, right, cancers for some people who has inherited an abnormal copy, they already start out with one mutation. And it's easier and quicker for those individuals to get the type of the cancer compared to people who have not inherit the disease version of the gene. So one such gene, actually we'll come back to the penetrance later on. One such gene is called the BRCA1 gene. Okay, I know we are time's up already, just 5 more minutes and then I'll be done. There's something called the BRCA1 gene, which stands for breast cancer 1 gene. This is responsible for DNA damage repairing and destroying the cell when you cannot repair the DNA. In other words, this gene codes for a red light. It stops the cell from dividing when there is a problem. Okay. So in the general public, women, like how likely is it for women to get breast cancer? Well, in the general public, only 12% chance of getting breast cancer sometime in their lives. However, for women who inherited one copy of the mutated BRCA1 gene, okay? In other words, for women who are born with one broken red light, right, because of BRCA1 gene, their chance of getting breast cancer is as high as 55 to 65%, right? That they will get breast cancer by the age of 70. So much higher compared to only 12% in general public, right? Some of you probably know who this is. This is Angelina Jolie. She's an actress. And her mother died of breast cancer, I believe. And so she went for genetic screening and found out that she inherited the BRCA1 mutation gene. Okay, so all the cells in her body has one broken red light. If you're following the mutation, the analogy here, so she is at risk of getting breast cancer, right? 55 to 65% chance. So to minimize the chance of getting breast cancer, she actually went and did a double mastectomy, which basically means I removed the breast. And that lower her risk significantly, right? Because now the breast tissue is not there. You can't really get breast cancer without those. That actually causes other people to want to do the same thing, right? They want to find out if they're at risk. And if they're at risk, if you could afford it, this is an option to significantly reduce your risk. But it's not an option for everybody. It's expensive, and some people live with the risk, and that's okay. But it's an option, right? And when I say it's just a higher risk, is because of this concept of penetrance. Okay, this is the last thing, I promise, and then we're done. Penetrance is the concept that describes how likely are you to actually get the disease if you have the disease gene. Okay, it turns out just because you have the disease gene doesn't guarantee you're going to get it, right? You might have a higher chance of getting it, but you might not get it for sure. Right? Sometimes it's complete penetrance. Complete penetrance means if you have the disease gene, you will guarantee to have the disease. Like that's like the worst case scenario, like a complete penetrance. Incomplete penetrance it depends. Right? Sometimes it's like 50% penetrance. Right? So only half the people with the disease gene will actually get the disease. The lower the penetrance, the less likely you will get the disease, right? Okay, so there's that factor to take into consideration as well, right? It's not so nowadays it's very common for people to, what is it called, like 23andMe or something. Like there's a lot of companies there that could screen your DNA and tell you about your ancestries and also give you a profile of your genes, right? Like you can find out if you have certain genes that makes you more at risk of getting certain diseases, right? But to me, like there's no point finding out because of the concept of penetrance, right? Like just because you have certain indicators does not automatically mean that you're gonna get sick from it. Okay. Of course, there are some conditions where it's more higher chances of getting it, which we'll learn later on. But for the most part, there's still a lot of wiggle room. Okay, that's it for today. We still have a couple more slides. We'll finish up next time. Now, for next class, please print the study guide for lecture 7. Because there's a lot of problems we have to solve together in lecture 7. It's kind of like your math class and chemistry class. So without the study guide, it's hard for you to follow. All right. Thank you for coming and thank you for staying extra time. Have a good week. I'll see you all next Tuesday. Bye-bye.